Hello and welcome to Exploratory Data Analysis Module 2, Leaves and Trees, where we'll take a little more in-depth analysis of this topic along with some examples. I'm Dr. Mark Williamson for DaCCoTA and the University of North Dakota. As a bit of introduction, the purpose of exploratory data analysis is to summarize the main characteristics. So in our first module, a bird's eye view, we looked at 4 different pieces of it, which I focused on viewing data, summary statistics, basic graphing, and basic tests. So what we'll be doing today is looking at a little more of the rationale and descriptions of what we're doing, as well as step-by-step examples and some assessments. Finally, we'll wrap up with some caveats, real-world examples, and summary. So let's get into the rationales. Really, why should we perform exploratory data analysis? I think of this in 3 big areas. The first is to get to know your data. So one, it can help you catch issues, like maybe your data is not formatted correctly, maybe you're missing data, so on and so forth. Maybe you even need to redo some of your data. So that helps you figure that out. Also helps you get your head around complex data. Like for example, this is just some descriptive statistics for a car data set. You can see there's a lot of variables going on here. And there's lots of ranges, like for example, 4 in cars. That's actually a binary. Either it isn't or it is. While other ones can be decimal, can be have a very high range, and in fact, not all have the same number of observations. It's hard to detect that without exploring your data. Also, it helps you with coding issues, especially if you're a first-time analyst. Maybe you're working with SAS, R, even Excel. You need to know your data so you can enter it correctly, because computers are very dumb, but they're very fast. So they'll only do what you tell them, and they can do things wrong very fast and efficiently, so it's up to you to correct mistakes. For example, here I have a model statement in, say, SAS, where we're looking at temperature as a function of weight. So really what that means is we're trying to use weight to predict temperature. But just as easily, we could put weight and temperature. Same thing in R here. Both of these, if you enter them in, SAS or R will happily run it for you. But if you get it mixed up and you're actually trying to do one rather than the other, you won't tell it unless you're paying attention. So that's another reason. Second part is it'll really help you save time and effort in the long run, so it's kind of to your benefit. First, it can help you plot out a future vision. So there's a lot of analyses, a lot of tests. Like for example, this is just a decision tree for certain types of test. Generically, it can help craft what you're ultimately going to do instead of trying a bunch of things that won't work. Also, in a related bent, it'll help you demo the results. So it could be something as simple as a box plot, where already we have 4 different types of cancer and cancer rates. Already we see that breast and prostate cancer are higher than colorectal and lung, so that's going to tell you something about what it should ultimately turn out. And you can actually use a simple representation to compare to your results to make sure you did it correctly. And then finally, it should help you reduce mistakes. It's very frustrating when you've spent all this time in analysis and you realize you did something wrong at the very beginning, so you have to go back and do everything again. Exploring it thoroughly, understanding it, helps reduce that chance. And then the third big one is it leads to defendable results. So a lot of what you're exploring is figuring out what to do, and because you know your data, you know why you're doing something, and you have a rationale for it, you can explain why you did what you did. Also, a lot of this exploratory data analysis, like these plots or tables, they give summaries of the data that you might be adding to results anyways. And then finally, a lot of these explorations, you're testing assumptions for certain tests to do so that you know your results are valid and you can defend them because you've explored and met the assumptions. Next, let's go through a few descriptions. If you're familiar with statistics, this should be pretty much a review, but just in case, it's good to have these in our minds. First of all, statistical models. When I talk about a statistical model, it's a mathematical description of how data can conceivably be produced. For example, we might have a pretty common model like this. Y is our variable we're trying to predict, we're trying to say something about, and here's the equation describing that. Sometimes this is known as like you can think of this more simply as like the intercept plus some sort of constant and another variable and then some error. And this helps describe how Y might be produced. Next, we have parametric data. It's also known as normal data or normally distributed. Essentially, it fits a normal or Gaussian distribution. And this is a big assumption for many statistical tests. So we almost always want to know whether it's parametric or not. And we can kind of look at that. This is a parametric or normal distribution. We see that it's centered around the mean and it's symmetrical on both sides. Next couple of things, paired and repeated measures. So paired data is like two measurements that are not independent. And that's really important because we work with that differently than ones that are. So a great example of this is like a before and after. Like maybe you measure someone's blood pressure before and after a workout. The before measurement for, say, individual one is highly related to the after measurement of individual one, so we have to take that into account in our analyses. And then repeated measures is sort of like an extended version of that, where it can be two or more. So maybe time intervals is a good example. Maybe you measure someone's blood pressure over 3 or 4 months, and then you do this for several individuals, and maybe you're looking at a change across time. And then finally, talking about variables, we have independent and dependent variables. Independent variables, they do not depend on another variable. And we also think of them as causative variables or predictive variables, or more simply like the X variable. And we use independent variables to try to predict or say something about dependent variables. They are our variable of interest, and they depend on other variables. We also know that the response variable or the Y variable. So up here in our equation, this is the dependent variable, this is the independent variable. All right, with all that out of the way, let's drop into an example. I'm going to start out by using R software, and I'll use the built-in datasets package and the dataset trees. Now this contains the diameter, height, and volume for black cherry trees. And so let's set up a research question to try to answer. Can we use girth or height to accurately predict volume? And we can see how this might be an important question because getting volume from a tree is probably pretty difficult, but getting something like girth, so getting the diameter around, or the height, just going up, is much easier. So if we could use these to predict volume, that'd be possibly useful for some sort of ecological purpose. So let's start by looking at the data. We can use print on trees. And here we go. We see that there's 3 variables, girth, height, and volume, and that they're all numerical. And there's no missing data. So that tells us stuff already. Let's do some summary stats, and we can do that simply in R with summary trees. And here we go. For each of the variables, we have the minimum, first, third quartile, median, mean, and max. And this is good to know this time. So it doesn't give us too much information because we really can't compare like across the 3. The fact that the girth is 13.25 and the mean of the height is 76, we can't really directly compare because they're measuring different things, but it does give us a little information here. Now let's look at graphing these. We can do a simple bar plot between the trees. And again, we're not comparing the ones against each other, but we can look at them individually. We see that girth is pretty well constrained. There's not a lot of variation. It seems pretty symmetrical. Height has a little more variation in it, but it's still quite symmetrical. Whereas volume, there's a lot of variation, and we see that's not symmetrical. This is the median. It seems to tend a lot more towards higher volumes, and we even see an outlier here. So let's look at histograms to kind of check normality. So for girth, it's not great, but it's not too bad, and height is quite a lot better. And really, when it comes to normality, we don't need to worry too much about the predictor variables or the x variables. We really need to worry about the y, like the dependent variable. And here we see, youch, that is not normally distributed at all. So one trick we can use to sort of finagle our data into approaching normality would be to do a transformation. So a pretty common one here I have is create a new variable that is the log of volume, and I'll show the histogram here, and bingo, there we go, it looks a lot better. So from now on, let's use natural log of volume, and we can do QQ plots with QQ norm and line to look at another way to test for normality for both volume and log of volume, and we see that, yeah, for the actual volume it doesn't really fit the line very well, though when we transform it, it gets a lot better. And then we can look at scatter plots of the variables. So for girth and natural log of volume, we see that seems like a pretty strong relationship positively. And then also positive relationship for height, though not nearly as strong. And then it's often good to look at our two predictor variables against each other to see how they're correlated or related, because in tests that we might run later, we might want to add both, but if they're too related to each other, they're not really giving more information, and so we usually avoid doing that. And we see that they're, pretty related. All right, well, we never actually got any numbers. We just looked at kind of visualizing relationships. So let's get some correlation numbers here by doing tests for the three variables. We see that, yep, like we expected with volume and girth, it's very highly correlated. So correlation, the numbers run from 1 to be like theoretically perfectly correlated positively to -1, which is perfectly negatively correlated, and then 0 would be like no correlation at all. So we see girth is very highly correlated positively, and then height is decent, though not nearly as much. And looking at the two predictor variables against each other, they're about in the middle there, about 50 or 0.5. So now let's do some simple tests. We're going to create linear models, so LM. This would be a basic regression where we have one Y and one X. So let's do this for girth. There's a lot that pops out here. Let's just focus. So here's our formula. Here's sort of the coefficient. So this is our X variable, and this is our p-value, which is much less than 0.05. It's actually approximately 1, so it's significant. And we see that multiple R squared is one way of showing how much it explains it, and we see that 1 would be the theoretical max, so it explains a lot. It's really good at predicting. Girth is really good at predicting volume. So let's look at height now. So we do, once again, create a linear model, and then a summary pops up this whole thing. Let's get rid of this stuff here. And we see that similarly, height is positive. It's still highly significant. Though we see the multiple R squared is a lot lower. It explains a lot less of volume, but it's still significant. So kind of my conclusion, all this step-by-step example would be that we're going to run some sort of regression, probably just a very simple linear regression with girth, because that seems to explain pretty much all of it. All right. So now let's go on to an assessment. Here's 5 questions here. I would suggest you pause this, work these out yourself, and then rejoin me in the video. Hello and welcome. Hopefully you worked on this and you're back down, maybe even got a snack, and you're ready to see how you did. So first of all, we're asking about the X and Y variable, so the dependent and independent for this R code where we have leaf number and branch number. And so leaf number is going to be the Y, the dependent variable. So we usually do something like, Y over on the left, tilde X, excuse my poor writing. And then the branch number, of course, would be X or the independent variable. Okay, next we're asking about which variable has the strongest relation to apples. So we're doing correlation tests, so fig, chestnut, oak, and here's the results. And what we find here is that oak has the strongest relation because it's the biggest number. Now it's negative, which means there's a strong negative. So if we do something like apple and oak, it's going to look something like this. And if we ran some statistical tests, a bit of a line like that. And 3, we have 3 different relationships. I'm asking, is there a relationship? If so, what kind? So A, it's going down. It looks pretty clear that it's a negative relationship. Where B is yes in a positive way, and there's really seems to be no relationship. So if we try to put like a trend line, it would be something like that. So really nothing. As X goes up, Y doesn't really do anything. Okay, and finally, with this histogram and ggplot, it's normally distributed. Well, if we kind of draw that normal plot, it looks pretty decent, and all these seem to line up pretty well. So I'd say, oops, I forgot about question 4. What 2 graphs? Well, the ones we see here, histogram and ggplot, and because of them, we see here, yeah, it looks like this data is normally distributed. Okay, great. Now let's switch gears a little bit and do an example in SAS for all you SAS users out there. So we're going to take a look at the SAS help library. It has a lot of data sets. We'll use dataset fish. It has weight, length, actually 3 different measurements. If we were using this in a certain way, we could actually do a repeated measurements trial if we wanted to. It also has height and width, and these are 7 different species of fish. So the research question we're going to ask here is there a width difference between the species of fish? So we can look at the data, start by using proc print to get our data set. And now here there is a lot of observations, so we cut it off at 20. What we do see here is there are 7 different variables. Species, seen fish here, this is bream. There's actually 7 categories, and they're nominal because you can't really order fish. And then these are all numerical continuous. And one important note we see here is that weight has a mission variable at observation 14, which is good to know. Now, it won't actually affect our analysis because we're just using width, and they have all the variables there. And so we're only going to use species and width, and we're going to ignore the rest. So then we can do some summary stats. We can look at fish and then their variable of width, so this column here. Do that first, and what we see here is pretty basic stuff. We have the mean, the medium, the mode, and variability. So that gives us a little something there. So the average fish is 4.4, whatever, ounces, pounds, whatever they used. Probably like grams. That seems a little low, but I don't know. We'd have to dig more into the background of the data. And then frequency. So since fish, they're not numbers, we can't give a number, but what we can do is we can look at frequency. So how many fish of each species. So we do that, use proc freq. And then what we get out here is the frequency, the counts of fish. You can see that perch, they had the most, over 35%, whereas whitefish actually only had 6. So that's actually really good to know. So we know that the different group sizes are very different, and that's often one assumption. So now let's do some graphing. We can look at the histogram of width overall. So we can just do proc sgplot, and hey, look, width looks pretty good. That's pretty normally distributed. Then we can look at like box plots of each species of their width. We can just do that here. And this tells us actually a lot of good information. And now, since they're all measuring the same thing width, we can compare the different boxes. See that some, like perch, they very variable. Other ones a lot more constrained. And we see here like smelt is definitely the smallest. And that whitefish was only at 6. We see, there's not a lot of variation that doesn't have the min and max, because that's essentially the quartile. So once again, whitefish is probably going to be an issue here. And another thing we're seeing here is the size of these bars are all very different, can all be very different, which means their variance, which is one assumption, is probably not going to be met. So we'll look at that next here. We can run some simple tests. So we're ramping up to do something like an ANOVA style test where we're saying, of these 3 or more groups, is some variable different at least one group? So does at least one fish species have a different width than the other? That's what we're going to try to answer here. So there's some assumptions there. There's assumption of independence that, like, for each sample we take a fish, they're not related to another. We really can't do any tests for that. That's essentially going to be in your design. You need to set up your experiment to ensure that. But then there's obviously normality is another assumption, and the third assumption is equal variance. So first, let's take a look at normality. Now, since we're doing different groups, we have to actually look at the width by species. We can't just look at width overall. So we're going to sort our data first, because that's usually what most of these, any procedure in SAS that has like a by group, you need to sort it by the same thing earlier. So we're going to do that, and we're going to get a histogram with actually a nice little, I don't even have to draw it, normal distribution for all 7 species. And we can see that they have varying degrees. Some look pretty decently normally distributed, others are more dicey, and this one, which I think is the whitefish, is not even close to normally distributed. So it's like, oh, okay, that tells us a lot that not all groups are normally distributed. Then now let's do equality of variance. There's different ways to do it, but what we're going to do here is we're actually going to run a model. So we're going to use this thing called PROC-GLM, which is generalized linear model. It's a more general approach to things. So it can do things like ANOVA, it can do more complicated things. So this is nice, this sort of multi-purpose tool. So we're going to essentially have to say species are a class, or they're not numerical. We're going to model with as a function of species, trying to say, if we know the species, can we determine the width? And then we're going to do this homogeneity of variance test, a Levine type. This is going to give us kind of our answer of whether or not there's equal variance. So first, this is just the actual generalized linear model. There's some stuff going on here, but really what we see here is it's significant, which means at least one of those 7 different species of fish has a significantly different width. And then we can go on to do this Levine's test for homogeneity. So what this says is the null hypothesis is that they have equal variance. And so if we have a significant p-value like we do here, the null hypothesis is rejected, meaning that the homogeneity of variance is not equal. So at least one group has different variance, and this is that plot again, and yeah, not surprising that this is a very small box, this is a very large box, this box is even larger. So, which means we can't just assume and do a standard ANOVA. What my conclusion of all this is, we probably want to run some sort of modified ANOVA. So there's some ANOVA tests that that actually don't assume equal variance, or we could do a more general linear model. And we might even want to think about taking out whitefish, since it has such a small sample size, if we can justify that. Okay, here's another assessment. It's probably time for you to pause this and work this out yourself, and maybe even take a nap. Who knows? I will see you back in a second. All right, hopefully you're back, full of answers and possibly renewed in energy. Let's see number 1. This is a similar question to last time, except for we're using SAS notation, and unsurprisingly, it's going to be y as length, the dependent, equals x as the independent. So we have a equal variance test. We're asking, is there equal variance? So we see here, this is not significant. So what that means is it's greater than 0.05. So we fail to reject the hypothesis that the variances are equal, which means we have no reason to believe that they're anything but equal. So yes, there's equal variance. 3, there's some box plots. We're asking you to predict whether or not they're going to have equal variance. And I would say no. I mean, these quartile lengths between here and first and third quartile, they're very, very different. And then how can the assumption of independent sampling be tested is, well, it really can't. You have to have good sampling design to make sure the assumption is met. And 5, we're asking, we want to do a test. It's fuel efficiency across 4 different car makes, so 4 different groups. How should you test for normality to run an ANOVA? We can either check the normality over all makes, so just do fuel efficiency, or normality for each make individually, so fuel efficiency by car. And the correct answer is B, fuel efficiency by car. Kind of like we did with the fish, we need to check each car's distribution. Okay, let's turn to a few caveats and concerns here. So the first one is, as I kind of suggested, normality tests are really an art. I mean, there are actually like tests you can run that'll give you a spit out a number to say whether it's normally distributed or not. I don't tend to like those very much because they're pretty stringent, and so a lot of data that I would suggest is normally distributed or close enough don't really pass that test, which is why I like to use histograms and QQ plots, which includes a lot more subjective sort of visualizing, looking at it, and kind of deciding. So there's no wrong way, but it's really up to you to decide and be able to defend your judgment. Another caveat is there's more than one way of doing things. I showed you one set of ways. There's certainly other methods, techniques, and obviously for more complicated or different examples, you're going to need to do different things. So keep in mind that it's often not a strict linear road. It's a road with a lot of paths that you have to choose yourself. 3, we saw by a lot of things, code output can be very confusing. R and SAS sometimes spit out a lot of things. So you need to be able to pay attention for what you're looking for and what the tests mean. Because like, for example, some tests, like that linear model test, a significant p-value was good or interesting because it says, oh yeah, there's at least a difference. Whereas that homogeneous variance test was, if the p-value was significant, that was kind of a bad thing. It was like, oh, which means my groups aren't have equal variance, which means I have to do a lot more work. So keep that in mind. Finally, data can be problematic both by nature and design. That fish example is kind of problematic because some of the sample size, like there's only a few whitefish, a bunch of perch. That really was kind of by nature, like they were pulling stuff out of the ice and they couldn't like decree that an equal number of fish come out. But you can also have poor or good design set to make your data cleaner or messier, and you always have to deal with the possibility of unequal sample sizes and unequal variances and all sorts of other things. So it's important to design things well, but also realize that nature is not perfectly clear. It's kind of messy, so you'll have to be able to handle any sort of data set as it comes. Data is rarely idealistic. All right, so let's look at just a couple of real-world examples of doing some data exploration. This is a great paper by Zur et al., which actually has a 10-step thing for regression type analyses that is really like a great go-to guide. And we can see how important data exploration is. It's the third step. And here's just an example they have here of beautifully visualizing pretty complicated data. So we have one Y variable clam length across a bunch of other categorical variables. So these dots show the clam length, but then across different birds. These are hammers or stabbers. These are kind of oyster catchers. But then they also have across 2 different months, so that's why we have these columns. And then, of course, rows. And then we have different observations. I think like observations A, B, and C, and so those are our columns. So that's a great example. Here is a paper on COVID by Dr. Ahmed and Dr. Mark Williamson. And this is just a way of visualizing median for a variety of possible variables that might be of interest across different regions of the U.S. And this was good for visualizing and referencing. And finally, here's a map from a cancer paper, and this isn't so much exploration as sort of setting the scene, where they're looking at North Dakota data, but then they compare it to the rest of the U.S. just to kind of frame the data that they'll be exploring. So that's, this sort of setting up the data isn't quite what we covered, but it is an important extended aspect. All right, so to wrap up here, we know that exploratory data analysis is a necessary first step, and it really helps us to know our data, save time and effort, and then really get good defendable results. And there's a lot of ways to get it done. We looked at 2. You can use whatever software of your choice, like SPSS, Excel. Heck, you can even do some of this stuff by hand. So thank you for joining us for this Leaves and Trees. Tune in next time for a plunge into advanced topics in Module 3, Deep Dive. Thank you.